the question about how inevitable is life, how inevitable is this planet, and how alone or not alone are we in our own solar system, let alone our own galaxy, let alone the universe. I think that what we really need to do is to be seriously bringing together fields that don't normally work together, or work together in a way that, but in an authentic way to address the problem. Life doesn't exist passively on this planet, and this planet doesn't exist passively with regards to life. And the two are intertwined in ways that we don't understand. And I think it's an intensely interesting area to study.
Hi, everyone. I'm Sarah Kaplan. I'm a reporter at the Washington Post. So even though tonight is about Earth's journey towards life, I actually want to start on Mars. In 1976, when the first successful spacecraft to land and operate on Mars landed, the Viking landers. And these spacecraft were they were equipped with this wide array of instruments aimed at finding out whether or not there was life on Mars. There was even talk of putting a light on these spacecraft in case there were little Martian critters that might be attracted to it. And we all know now that Mars is a desert without oxygen in its atmosphere and no life that Viking could detect. And I think that looking back now, there's a lot we know about life that we just didn't know in 1976. I mean, the year after the Viking spacecraft, that there are two of them, the year after they landed, scientists went deep into the ocean and found just a wide diversity of organisms living around hydrothermal vents and living off of chemicals in the ocean coming out of those vents. We found life in clouds. We found life deep in caves where there's no light. You're going to hear tonight about life that just should challenge all of your assumptions about what life even is. And I think that if we're going to go searching for life elsewhere, we have to understand life in our own backyard. And so that's what tonight is about. We've got a brilliant panel, and I'm going to read their names because I want to make sure I get them in the right order. So we're going to be looking at this planet, what has happened in its past, what we can observe in labs today to understand what life is and why it flourishes here on this planet that we are all very lucky to have found ourselves on. And hopefully that will better equip us to find it elsewhere. So with that, I want to introduce Marilyn Fogel from the University of California, Riverside, Patrick Shih from Lawrence Berkeley National Lab, Devaki Baya from Carnegie, and Arthur Grossman, also from the Carnegie Institution, Jinko Wang from the Whitehead Institute and MIT, and Andrew Steele, also from Carnegie.
I've been fascinated by this since I was a college student when I took an earth science class, and the first papers on finding microfossils of algae were found in Precambrian rocks, which were maybe 1 billion years old. And it's fascinating to me to look at those really old rocks, and now I've been in the field to look at them and see how extensive they are, going for kilometers and kilometers, all made by microbes. That there was a system on the Earth that had nothing to do with our built environment, but just was formed by what we would call simple, but we know now they're not simple microorganisms. That's fascinating to me. So the origin that I'm going to talk about today is the origin of ecosystems, and we're starting big on this topic. And one of the things that strikes you when you go out and you look on the Earth is that we are never alone. Wherever you are on the Earth, you're always in a place where there's a concert of organisms. What you see on the slide here is on the top of a glacier in Svalbard, which is about 80 degrees north latitude. And you're looking at sort of pinkish snow, where snow algae are colonizing these very extreme environments, as well as bacteria. When you go back in time on the Earth, one of the big questions we have was when in time did life originate on the Earth? And what we have to do this, to understand this, is to go out and collect rocks and analyze them chemically to see what we find. What you're seeing on this slide is you see those circles on those diagrams. Those have been formed by, these go for kilometers, miles of a distance. They were formed by single-celled organisms, cyanobacteria and other bacteria called stromatolites. So what we want to do in our research is to determine whether something was made by biology or by non-biological processes. What you see on this slide is the result of algae, cyanobacteria, precipitating calcium carbonate in a thermal spring. And the outcome of that is a combination of both living and physical processes. So when we go to look for origins of life, we're looking for this intersection of geology and biology taken together. This is another photograph that we took when we went to Svalbard. You go on a big icebreaker ship, and you're looking at the landscape. Not only is it gorgeous, but you're looking at that green. What is that green? That green is mosses and very simple plants. And you start to come up with ideas, what made these patterns over there? And after a while, I'm looking at this, and I'm the ecology on a NASA expedition. It turns out that's the interaction of seabirds. This is a picture of a seabird over there. Seabirds foraging on the ocean, coming up to these mountains, nesting over there, excreting, and turning those sort of desert ecosystems into living, breathing, very highly biological systems. And that pattern, that interaction, this is an interface between the ocean and the land, is definitely something that I think is a sign of life. This slide over here is showing you a thermal spring, also found in Svalbard. And you'll hear from my colleagues over here that these are springs colonized by thermophilic bacteria and algae. They form mats. There are mosses in here, as you can see over here, and a very complicated ecosystem, so that when we go out in the environment, when we're looking for origins, we're not looking for a uniform pattern. We're looking for how organisms relate to one another. And that is really the crux of how did we get from something that was squish and nothingness to complex life.
So Marilyn, you talk about how organisms are the product of their environment and of their ecosystems, of the other organisms around them. And I'm hoping, as our resident geologist on the panel, that you can tell us a little bit about sort of early Earth and that environment. It was very different from what we know today. So how might those conditions have shaped the way life arose, and maybe even were ingredients for life delivered from elsewhere?
Well, the early Earth, the atmosphere was very different than it is today. Today we have about 20% of our atmosphere is oxygen, which you know about, and about 400 parts per million carbon dioxide, which we worry about, right? And in those days when the Earth was created, we primarily had, we had little to no oxygen in the atmosphere. There was several percent of carbon dioxide and mostly nitrogen. And so the world was anoxic, anaerobic, no oxygen. There was a lot of iron dissolved in seawater. We had the faint sun. The sun was not as strong as it is today. There were times when it was really hot, really cold, and it was actually before plate tectonics arose. And it was a time also when you had the heavy bombardment of asteroids and meteorites coming in here. And one of the things that people have considered, I don't know whether I go along with it, was that organic matter that is found in a certain type of a meteorite called a carbonaceous chondrite brought some of the amino acids and nucleic acid bases to the Earth, and perhaps, it's a little murky, whether they formed some of the early compounds of life. But through time, this changed, and we entered into over several billion years. I would say 2 billion, 2.5 billion years. Things changed to more like it is today.
Well, I think actually that's a good place to sort of open it up for your questions.
You were discussing earlier about the difficulty between distinguishing evidence of fossils of early life on Earth versus physical processes. And I was just wondering, how do you go about constraining what those physical processes would be in the rock record when they were so different in the early Earth as well, pre-plate tectonics and things like that? And then simultaneously, how would you then extend that to other extraterrestrial bodies, those physical processes not necessarily being what we see in the, post 2.5 billion year Earth?
So I could start on one of the things that I have absolutely no experience with, but my colleagues, my former colleagues at the Geophysical Lab, is to look at the petrology of the rocks where you're finding these fossils. And Steely would agree with me here that the more attention you pay to what minerals are found in there, what their history was over geologic time, you're going to have a much better idea as to whether these are original things that you're looking at or subsequent contamination. You want to jump in from there?
Context is key in these measurements. Like we know from carbonaceous chondrites that are filled with carbonaceous material that probably wasn't generated by life. And if you look at organic chemistry and well over a century of the reactions that take place in organic chemistry, you can find places in the environment where organic chemistry happens as carbon dioxide, carbon monoxide, methane come into contact with catalysts. So you see organics being produced abiotically in the environment today. There's Rainbow Ridge being one, hydrothermal hot springs. And so the most important thing is to get the context right of where the sample's coming from, to understand its history, to understand the petrology and of the rocks that are making it up, and then to look to see where that carbon is or where those organics remains are, and look to see what's around them. Are they associated with known catalysts? Are they just sat there? What kind of morphology do they have? What kind of isotopic signal do they have? What kind of molecular makeup do they have? And so you can use all of those tools in a stepwise way to understand where the sample is. The problem comes in the really early life debate is that a lot of those samples are out of context, and a lot of the basic petrology was rudimentary at best. And so that has generated a generation of debate on samples that we really need to go back and address that. It was a good first start at the problem, but due to the problems and the debates that have come from looking at those samples, we've learned that actually probably wouldn't do it the same way again, right? And so we go back and check. We need to go back and check using that kind of flow, scientific flow, to looking at the samples again.
For a long time, our understanding of the early Earth and early life has been very descriptive. We've looked at the fossil and rock record, and we've come up with a lot of hypotheses about how life may have evolved, but there's a huge amount of unknown about what's gone extinct, what aren't we capturing in the fossil record or the rock record. And because of that, pretty much anything that we could imagine might have existed at some point, and really trying to make sense of all of it is what keeps this field fun.
I'm interested in three defining features of our planet: the presence of water, the presence of oxygen, and the presence of life, and how they're all intertwined through the evolution of photosynthesis. And really, if we can begin to better understand when and how photosynthesis evolved, we can get at the core of some of the major questions that we have, such as how did life originate and evolve, and also how did the early evolution of microbial metabolisms alter the trajectory of our planet? So in order to ask these questions, we need to have a better grasp of geological time, which can be very challenging and abstract to think about sometimes. So really, everything that you can see with your naked eye all evolved in a tiny sliver of time on this timeline at the very tail end. And we know that the early Earth, it was very different from today because there was no oxygen around it. Oxygen began to accumulate in low levels in our atmosphere around 2.4 billion years ago. So we can loosely think of Earth history because of this in two different parts, before oxygen and after oxygen. And we know that life was around before this major transition. And one of the major questions that we've had is, how did anaerobic life before this event evolve to produce oxygen and evolve to survive in these oxygenated environments? The short answer is through photosynthesis. And arguably, photosynthesis is the most important biological innovation that life has ever evolved. And that's because it enabled life to convert light energy into chemical energy by splitting water into molecular oxygen. And this is the oxygen that we're breathing right now. Now, that chemical energy was then used to fix carbon dioxide out of our atmosphere into organic compounds and sugars, which drive our global ecosystems. So usually when we talk about photosynthesis, you normally just think of plants. But actually, a huge chunk of global photosynthesis is actually done by these little critters called cyanobacteria, which come in many different colors, shapes, and forms. We actually know that cyanobacteria were around way before plants, and therefore they were the original inventors of photosynthesis. So one of the major questions that we've had are, how did these ancient or extinct cyanobacteria first evolve this ability to undergo photosynthesis? And the short answer is we really don't know, because much of our understanding of early life stems from the fossil record. And because these cyanobacteria are so small, it's very difficult to find fossils of them to understand how they originated. My research is interested in not looking for these tiny fossils, but rather to try to leverage synthetic biology to reconstruct aspects of ancient life. So instead of going out and looking for dinosaur DNA in amber like they did in Jurassic Park, I'm interested in finding as many genes as possible and then using computational methods to predict what did ancient genes and ancient proteins look like. And then from there, we can use synthetic biology to actually resurrect and reconstruct these ancient proteins in the lab and try to characterize them, to learn aspects of early life and the early Earth. And really, we might be able to shed some light onto how we might want to look for life on other planets, because the early Earth was very different from today, and therefore may serve as a functional analog of what life may look like on other planets.
Patrick, I wanted to ask, synthetic biology, the idea is you're sort of reverse engineering those like primitive organisms. Can you kind of like walk us through what does that mean? How would you reverse engineer something that probably has been extinct for hundreds of millions of years?
Yeah, so pretty much we try to take as many present-day sequences of genes right now, and we know that we can reconstruct their evolutionary history. And by doing so, we can actually just use probabilistic models to try to make a prediction as to what these ancient sequences would be, which would predict the evolutionary history that we see today. So this is a really important technique because a lot of these questions that we want to ask are just fundamentally challenging. And to be honest, we probably won't find the answers to how did life originally evolve. But we need to start to think outside of the box and think of alternative methods. If we can't find these fossils, or if we can't go to the rock record and look for things, we have to try to come up with testable hypotheses, which is where synthetic biology comes in. Thank you for a really fascinating evening. We know a lot about the physical environment on Mars, the spectrum of radiation, the abundance of materials, the presence or absence of water or other ionizing solvent, and we know a lot about the environments, at least a little of Titan and Enceladus and some other large bodies. Given what we know about these places, are we aware of fundamental physical reasons to suspect that life would evolve in these places along a different trajectory from how that's happened here on Earth? So I don't think we know if it would be on a different trajectory, because even on our planet, because we only have the sample size of one trajectory of life, it's not even clear if we could rewind the clock of life, the tape of life on Earth, and let everything re-evolve, if we would get the same trajectory that we saw today. So I think the role of just historical contingency and just how much just rolling of the dice there is in these sorts of evolutionary trajectory is something that is of active research in the field of evolutionary biology. And I think really we don't even fully understand how that would work on Earth. And therefore it'd be very difficult to try to predict what it'd look like on maybe Enceladus or something like that.
I mean, from my personal opinion, I think it's good to turn that upside down. If I want to look for life on Mars, I assume there is no life, and I go looking to, disprove that hypothesis, right? And if there isn't life on Mars, why isn't there life on Mars, and what does it tell us about our own planet? So that's where I'm starting to come from this. But my feeling is the conditions on Mars at 3.6 billion and conditions at Earth were roughly similar, but with some very notable differences. Things like plate tectonics, active magnetic field, which is extremely important for holding on to an atmosphere. So you can get the exchange between the lithosphere, the rocks, water, and the atmosphere, protecting all of this chemistry that's going on. And forming these niches that allow that chemistry to continue for a period of time. Now, Humphrey Davy named the core the infernal engine. Mars's infernal engine stopped a long time ago. And if you think that, if you imagine the path to life is like 10,000 test tubes, and of that, 1,000 of those test tubes need a Bunsen burner, it seems that Mars got to kind of test tube 10 or 12, and then the Bunsen burner wasn't there. And that's my feeling. I haven't got. We have some observations to show that, but that's roughly where I'm coming from. So the path of life or path to life was closed down early on Mars. But if that is true, we still have organic chemistry going on Mars. Those reactions are akin to early Earth, and those reactions on the early Earth spark the trajectory of life on this planet, and that's why we need to go and look.
The idea that biology was sort of static has completely gone away, blown out of the ground. Genomes change, ecosystems change, interactions change. And so for me, that's just something that's a bit of a revelation, really. We can start thinking of life not in the ways that we've gone out and found, but build predictions of what we can do with life. And that might allow us then to say, well, it doesn't have to be this way. We can plan it to be different, and that might tell us more about what early life might have been or will be on other planets. There's a perfect way to start this, because some of you were at the origins of the planets the last time, and Stephen Hawking said this wonderful thing. He said, however bad life may be and seem, there's always something you can do and succeed at. And where there's life, there's hope. And I wanted to sort of turn that around and say, where can we hope to find life? And that's what I'm going to show you in the next slide. And the title is sort of Diversity, Dynamics, and Defense, which is wonderful alliteration, but it's more or less true. And we started this work about 20 years ago in the hot springs in Yellowstone, and we really focused just at those edges of the color where life begins in a way, where the microbes are doing their thing. And they're doing it at 70 degrees, it's cyanobacteria, and they can do photosynthesis at these tremendously high temperatures, and we really actually don't understand how they do it or how they hack it. And so what we've been trying to do is we got a group of again, molecular biologists, evolutionary biologists, to answer the question, can we take the genomes of these organisms and try to understand how they survive in these very harsh environments? And maybe that's a proxy for early life. Here's where the first biggest surprise was. We sequenced one of these genomes, and it's just a circular genome, vanilla flavored, nothing exciting about it. But then we sequenced the second genome, and we expected it to be more or less like the first one. And here's what we found. Just completely scrambled genomes. But let me explain a little bit more. What it is that each of these genomes have more or less the same core content of genes. They have to do photosynthesis, they have to do respiration, but they're constantly shuffling these around in their genomes, and they're bringing in genes and pathways from elsewhere, sometimes aided and abetted by viruses, actually, it turns out. So you have this concept of the pan-genome and a core genome, and it's really astounding to us how little we understand of this pan-genome. And so what I want to leave you with in the next couple of slides is to explain how we thought of populations, microbial populations in various niches, shown as squares. That's anything but that. And you can have genomes in all of these niches, and over time, that black arrow shows you their selection. So if, for example, the light green one doesn't do so well, over time it gets washed out, and the dark green guys do well in a particular niche. It's kind of a static and beautiful and simple picture. However, what I would like to argue today, or sort of suggest to you, is that if you have this whole very dynamic set of populations, then in fact they can exchange genes. That's why all the genes are different colors, and they can then evolve to be better in some kind of micro niche. And that's, to me, a sort of revelation, because I hadn't thought of it as such a dynamic process. And it raises a lot of interesting questions about how we want to, what do we call a bacterial species, which is how we began asking this question. And just to put another wrinkle on it, it's not that you ever study bacteria in isolation. We have, as microbiologists, for 150 years. We take them out of their environment, and then we say, aha, now we understand them. And here's an example. These are cyanobacteria that are doing phototaxis, and the light is coming in from one direction. As individual cells, they sense the light, and they start to make these beautiful fingers towards it. That's when they're all by themselves. They make beautiful patterns. We work with physicists to understand how that happens. But that's not how any organism, you, me, or microbes do it. They live in communities. And what you see on the other side is a whole community. They're thin, long guys who can move. The cyanobacteria, the little black sausages, move along those. It's a very complex structural and metabolic process, and it's changing. And that's the message I want to leave you with, but I don't do it as well as a microbiologist who said about 150 years ago that they're considered to be the most simple of all existing organisms because they're the smallest. But small and simple is not the same thing. And I want you to leave that message, and when you go out and take a breath of oxygen, think small, think respect, think cyanobacteria.
So your talk really challenges the sort of paradigm of how we think about organisms. I mean, so high school biology class, you think of, here's a giraffe. It is a single individual organism. It is a member of the giraffe species, and so it has a giraffe genome. But you have seen that, bacteria, at least, they don't work that way. They are trading bits of their genomes. They're living in communities that it's not necessarily that meaningful to study them in isolation as much as it is to study them in groups. How does that, if we're asking this question, what was the first organism, or what did the earliest life look like, how does this kind of complicate that?
Do I have 5 minutes or an hour?
You've got 1 minute.
The 1 minute is, I think, for me, and I think maybe for the population, folks who are looking at the gut microbiome, etc., are realizing that really studying things in isolation is tragically leaving out complexity. But the flip side of that, we don't have the tools, either experimentally or theoretically, to understand what are the rules that govern these communities, and I'll leave it at that.
The amount of information that we can get now through sequence analysis, through looking in different environments, is giving us new insights into the origin of life and where organisms came from. I think as we learn more about how early life has evolved, we're also learning more about how life is evolving now on Earth as the environment is changing. And I think the lessons in the past will bring us into the future with more and more knowledge.
What I thought I would tell you about today is a little bit about an organelle called a chloroplast. And of course, chloroplasts are compartments or organelles in plants that do photosynthesis. They're what allows the corn and the rice to fix CO2, make polysaccharides that you eat and that support the world. It started out not as a chloroplast, but actually it started out as a cyanobacteria, the chloroplast. And so I would echo what Devaki said. You have to have a lot of respect for cyanobacteria. And if you look at this slide, you could see that at the bottom of the slide, there's a non-photosynthetic organism. It's a protist, and it's hungry. And what it did is it tried to eat the cyanobacteria. But in fact, the cyanobacteria escaped being digested by that protist, and it maintained itself in the protist. And the protist at some point realized that it was getting fixed carbon or polysaccharides or sugars from that cyanobacteria. And the cyanobacteria was being protected from the environment by the protist. So this integration of two organisms stuck. And that's the first part of the, it's a very simplistic way that I put it, but it's the first part of the evolution of a higher plant that has a chloroplast that does CO2 fixation. Now, when that happened, there were a lot of problems that happened along with that. It's true that the cyanobacteria was in this non-photosynthetic organism, and it was absorbing light energy. But the metabolisms of the two organisms weren't coordinated. They had never been together before. And so that light energy was coming in, and it couldn't be used as effectively because the cyanobacteria couldn't effectively transfer as much fixed carbon as it could make. And so a lot of the light energy was being used to generate reactive oxygen. The light was exciting pigments, and those pigments were reacting with oxygen, and you get harmful products from that. And so the organism was hurting very much. That's the way it happened in land plants, and it's a very basic picture that I'm giving you. But we could see it in organisms where this particular type of association occurred much more recently than in land plants. In land plants, it occurred about 1 billion years ago, or over 1 billion years ago. In this particular organism that I'm showing you here, which is called Paulinella chromatophora, the same type of engulfment of a cyanobacteria occurred, but it occurred 100 million years ago. And so it gave us a chance to look at this type of evolutionary process over a shorter time period. We had a sort of glimpse, a window into the evolution of this photosynthetic organelle. And on this slide, you could see, in fact, that green compartment within the protist, if you do a lot of sectioning of it and you look at the ultrastructure, it's clearly a cyanobacteria based on that ultrastructure. And it's also losing a lot of its DNA, just like a chloroplast lost a lot of its DNA. Now, in losing that DNA, it lost a lot of its biosynthetic capacity. It lost a lot of genes to make amino acids, so it couldn't make amino acids anymore. It lost a lot of genes for photosynthetic function. It couldn't make those proteins anymore. It lost a lot of genes for cofactors like vitamins. However, it still does all those functions, even though it lost those genes, which means that the proteins, the genes are going outside the chloroplast, what's becoming a chloroplast, into the nuclear genome of the host organism, a lot of them, and it's being transcribed, translated, and the proteins now are going back into the chloroplast. Now, it's not only the genes that come from the cyanobacteria that are doing this, but it's a much more complicated process. In fact, this protist is taking up genes from outside because it's a heterotroph initially. That is, it eats other organisms, so it eats other bacteria. So it's getting genes from other bacteria. It also has its own set of genes, so it's retailoring its own set of genes. And if you look at the biosynthetic processes and the protein, the metabolic pathways that go on in the chloroplast, they're made up of proteins derived from bacteria through horizontal gene transfer. They're made up of proteins derived from the original endosymbiont, the cyanobacteria, and they're made up of proteins derived from the actual protist itself. So it's really a mosaic of proteins that make up these photosynthetic organelles. And that was a big surprise because we didn't realize that the genes have origins from all different types of organisms that the protist finds itself associated with. And that also gives us the idea that maybe in looking at this and understanding this, we could begin to tailor those genes ourselves and alter metabolism in some of these organisms to engineer them to make various products that we may want to use later on. So I think I'll stop there. Thank you.
So Arthur, we've been talking a lot about photosynthesis and the cyanobacteria, which are not to be underestimated. And I'm wondering, sort of, the first organisms that lived on Earth weren't photosynthetic. That was something that had to be invented. So why is understanding photosynthesis so essential?
Many of the first organisms that lived on Earth, they could use electrons from various sources and use energy like thermal energy and other forms of geothermal energy to take those electrons and raise the energy level to make carbon compounds. You will run out of a lot of the compounds that they used, and the most abundant compound to use is water. And if you could take electrons from water, you have an infinite supply of electrons to do biosynthetic processes. So it's critical to understand how to develop this type of capture of electrons using light energy, which is also very, very abundant, to understand basic biosynthetic processes and electron transport processes, even though these electron transport processes and biosynthetic processes are somewhat different than occurred in the very early Earth.
Research really into at the very beginning of how life emerged on Earth, I think has huge implication on how we understand life can continue to evolve on this planet. And we're actually learning quite a bit of information by studying the existing organisms, and also reverse direction, coming back to the origin and see how things could have come about at the first place.
So research in my lab is really kind of inspired by this remarkable biodiversity we see on a daily basis. So I was born and raised to really love plants. They are very diverse, and they migrated from an aquatic environment to land about 500 million years ago. And a lot of things have happened since then. And one thing about plants is they are known as so-called sessile plants. So once they're seeded, they're really disadvantaged. They have to deal with a lot of the abiotic and biotic stresses by doing something. So the thing they evolved to do is to become very good chemists. You can imagine you are a piece insect or you're a herbivore. What you want to do is just eat a lot of plants. So what plants do over hundreds of million years is to really elaborate their metabolism, coming up with millions of molecules. Many of them really to toxify anything that would eat them. And some of these become attractions for pollinating insects or seed dispersers to help them to transport pollen or seeds. So that actually creates a tremendous difficulty for people thinking about evolution. So Charles Darwin kind of proposed, how do we understand a very complex eye evolved? So this is known as a complex traits. So there are many things that have to happen to make an eye that can see. So I would argue a plant natural product, a plant molecule, as the one that you see on the right. So this molecule is known as taxol. It is made in the yew tree, and it's a very complicated molecule. Nowadays, we use it as a chemotherapy drug. And there are many bonds. They're too complicated. You can even very hard to put in that small space. So I'm going to tell you that every single bond is actually installed by a very specific enzyme. So to make this molecule, the plant has evolved more than 15 enzymes to put together this pathway to make this. And this one pathway only exists in one species. So it is really challenging to understand how this complex trait in metabolism could have evolved through a Darwinian process. So in the lab, we've been very busy studying very interesting chemical processes in plants. We're very interested in medicinal plants, so I'm just kind of highlighting 4 of the plants we study. The first one is a Tibetan medicinal plant producing a natural product that can improve blood circulation in the brain. The second one, the kava kava from Polynesian islands, used by local people to calm you down. So very calming anti-anxiety compound. The third one, the stinging tree, Gimpy Gimpy, produce a very remarkable cyclic peptide. If you got stung by it, the pain can last for months, if not years. And then the last one, the honey goat's root, is used in traditional Chinese medicine for cure sexual dysfunction and heart disease. So you may ask why you study those. So by understanding how plants evolved to catalyze interesting chemistry and making interesting natural products, we actually learn how metabolic systems evolve over time. And many of these catalysts become tools that we can use as synthetic biology tools to build future drugs, materials, and commodity chemicals that can sustain the 21st society. Thank you so much.
Jinko, what you were saying about sort of this question of how does something as complex as an eye or these really complex compounds that plants have developed, how do we figure out how that evolved sort of through Darwinian evolution? That's kind of the same big question we're asking right now, is like, how did all of this, all of us, the trees outside, come from what must have been just squidgy little organism, 4 billion years ago? So how do you think, you've looked at the evolution of different complex traits that plants needed to adapt to life on land. How does, how does that sort of connect back to this sort of idea of how life developed the complex traits needed to exist in the first place?
Yeah, so one of the questions that fascinated me since a child is the ability for the plants to migrate from aquatic environment onto land. So this is a remarkable event that happened on the planet. So before that happened around 500 million years ago, and you have to imagine, before that event, it was very barren land. There's nothing on land. And the plants have to really evolve many different traits to be able to live on a barren land. So one of the process is the evolution of the so-called phenolic pathways. So because you're shielded on the water, and the UV light is really shielded, but when you come onto land, you're exposed to UV. So the plants have really elaborated this pathway to make all these aromatic compounds that can absorb UV. So that process, if you compare the genome of any land plants with aquatic plant progenitor, you do not see that pathway. And that pathway can contain more than 20 enzymes. So that really challenged you how you make this elaborate pathway through this evolutionary process. I think that's a more tractable problem than trying to figure out what's the first metabolic system that early life evolved. So we now have a more workable system in plants, and by understanding this, maybe give us some hints about how early metabolism evolved in early life.
My question is it just chance that some molecule started replicating itself somewhere, some time ago?
So I can offer some of my insights. I think it's hard to say whether there could be another set of genetic codes. Now we use nucleic acids as the genetic basis for replication. I would predict there might be, but this is what the life now decide to do. But using some other cases, so early in the day, we've been debating whether chlorophyll is the only pigment that can be used for photosynthesis. But then the conclusion was there are actually many other forms of pigments that have been utilized to do photosynthesis in other forms of life, not in plants. So that implies there are alternative chemistry that can be utilized for some function. So I think that function can be the genetic code for another form of life.
We have an n=1 on life in the solar system, and that is this planet. And being able to deconstruct how that life started on this planet gives us a much better chance of finding life elsewhere. And vice versa. If we find life elsewhere, it gives us a much better understanding of how life started on this planet. If you look at origin of life studies traditionally, historically, there are a lot of worlds. People talk about protein worlds, metabolism worlds, RNA worlds, and we're talking pre-cellular here at this point in time. There's a lot of these worlds, and each of these worlds is studied by a certain group of researchers who have their interest in what happens in these worlds. There's only one outcome to this, and that is a system that is integrated. It is not just RNA or protein or metabolism. It is actually one complete organism. And so how did we get to that in the first place? Well, to my simplified reasoning in this is must have been a feedback loop early between the information system, which is your RNA and DNA, and the proteins. Because both of them, one codes for the other helps the other one survive, right? There's this link, this ecosystem amongst these molecules to actually get the job done. So whilst there's no universal definition of life, there are certain, and I'm going to introduce this concept of proto-life or proto-life molecule constructs, like viruses, virions, and prions. You might have heard of prions from Alzheimer's or Scrapie or CJD, these kind of pathogenic diseases that cause these plaques to form. Now, how many people in the audience here think that viruses are alive? Right? I don't know, about a third. So it's a third to two-thirds. I've even seen, this was a question given to me during my degree, are viruses alive? And I had to write an essay on it. The scientific community cannot agree if viruses are alive or not. If they're not alive, then what are they then? Minerals? Vegetables? No. I mean, there is no universal definition as to what they're doing as far as life goes. But if you actually look closely at viruses, now I'm going to talk about the properties of viruses and prions from now on, just the properties that interest me and may be applicable to the first organisms. One is self-assembly. Viruses make themselves. They put themselves together. Some of them contain their own genes for replication. Some of them are membrane-bound, and I'll go into that in a minute. They show evidence of being extremely ancient. They have been involved, as you've heard from my colleagues here, they've been involved in evolution and transferring genes for most of the time that life has been on this planet, around about 3.5. Okay? And in some cases, they do have a modified genetic code. They don't stick to the normal genetic code. This is a fantastic study by Cello in 2002, where they basically took a virus and they deconstructed the whole thing, and they took out its nucleic acid and they threw it away. And then they got a bunch of chemicals off the shelf and constructed the genome of that virus, put it together with the lysate of the cell, and it built itself up again and became infective. That is frightening. You have a bunch of nucleic acids and a bunch of proteins coming together to make an organism, or not, that is infective, that actually replicates itself. These are the simplest systems that show how replication started. But a virus is that simple, usually a protein coat and a bit of nucleic acid. But we're finding now, as we look more and more through these techniques that we've been discussing, molecular biology, and what I like to call molecular paleontology, where they're looking back through time at these genes. Prions are really interesting to me because basically they are incredibly robust. It's a protein that can survive 120 degrees C. Some mouse prions, you can set them on fire and the ash will still be infective, right? That's a selection advantage. There was a poor unfortunate series of patients who got infected from someone with CJD right the way down the line after autoclaving. And that's how they found how robust these things are. They bind to nucleic acids, they like RNA, they display Darwinian selection, they are they're inherited by the progeny in a non-genetic fashion, if you wish. And the difference between, so a prion is just a misfolded shape of a normal protein that becomes, in some cases, a disease, but in yeast, it can confer beneficial traits. It's not just about disease. In the last 20 years, we moved away from medical microbiology, where everything we know about microbes is because of how they infect us or kill us or whatever, and moved into the environment where we're finding a huge diversity of things from prions and viruses that we knew only as disease-causing agents. We're finding that they're having a lot of other functions now, and actually prions have been now found in plants and bacteria. So the activity of the protein on the left, the mainly green twists, denatures at 60, but the one on the right, which is the same amino acid sequence, won't denature at 120. And if you have a look, if you do a prediction algorithm on the entire protein database, what you find is they're ubiquitous throughout all domains of life. So what you have is a way of conferring a selection and survival advantage. If a prion binds a nucleic acid way back in a primordial pool, it's going to protect it. If it binds strongly, it'll protect it. If it binds loosely, it won't. But if it binds strongly, does that mean it's the origin of the genetic code? That strong binding confers a survivability trait. So is this where the origin of the genetic code came from? So in conclusion, the properties of viruses like self-assembly and prions like protection, binding, are these clues that we're finding to those properties that led to the first bacterial cell or microbial cell. These do appear in the most ancient of metabolic pathways. We can't decide if they're life or not. And if I get to Mars and find a virus, have I found life or not? Right? That's why I come at this and have this different kind of viewpoint on it. And the selection advantage may have allowed stabilization of protein-RNA interactions. The ribosome is a protein-RNA interaction. Right? Was the ribosome a virus? The common view is that a virus needs a host to survive, but we've already seen that there are viruses in the environment that don't. And my personal favorite about prions is the present in Saccharomyces cerevisiae. So every glass of beer you drink, you're drinking a yeast prion. Thank you very much.
So I've got a question for you that I think some of the other folks on the panel might also have an answer to. But it seems to me at least, if we're going to look for the first life on Earth or life elsewhere, we should probably know what we are looking for. So if you had to come up with a working definition for life, what would you say it is? And I guess also, if you think that is not important, like maybe we don't need a definition for life.
Well, so the philosophy that I've been using or through my work over the years on Martian meteorites and Mars missions have come up with is that you need only assume two things. It's carbon-based, and it doesn't use all the carbon compounds available to it. In the Murchison meteorite, there are over 10,000 different organic chemicals, but life chooses only a subset of amino acids, a subset of fatty acids, for instance. So if you know your organic chemistry and you're really good and know the context, all you need to look for is a signal outside of what you would expect from chemistry, right? Because life shows us again and again and again what it's really good at and what chemistry is really bad at is it speculates to accumulate. It will spend energy to gain energy further down the road, where chemistry is really bad at doing that. So you can predict the organic chemistry in any particular environment, and then you just look to see how that's perturbed to your expected. And then you don't have to define life. And it may not be ACT and G that is concentrated. It might be LMNOP. You don't know. You have no preconceived notion of the metabolism that you're looking for. That's how I'd go for it. Now, at some point in time, you're going to have to decide, is that concentration life? And I'd like to resolve the virus debate before we do that.
Steve Marin, I'm an astronomer, and this question is due to something Andrew said, but several others have commented on it. I'll give the astronomy example. When the space probe was launched towards Pluto, Pluto was still a planet. And there was an angry vote by astronomers who said it wasn't. But we got there and we learned all these amazing physical and chemical things about Pluto. They were the same properties whether you call it a planet or not. I wonder why it matters whether you define a virus as life. We have to study them. We get the flu. It'll matter maybe for which agency or department funds a program on viruses, or who gets the big prize for discovering life on Mars if you only discovered a virus. But is there a scientific reason you have to decide that question?
Me personally, I don't mind either way, but I tend to lean towards that they are like these proto-life type constructs. And I think that the properties that they show are really informative of everything from current evolution all the way back to to potentially the formation of complex life. But I agree, the definition doesn't really matter. It's like the Aliens film, isn't it? I don't need to know what they look like, I just need to know where they are, right? So that's how I approach the problem. I don't really mind what the definition is.
We also talked today about whether or not, what might be the oldest evidence of life on Earth that scientists agree on right now. There's a lot of disagreement. And I'm curious what you think is, what would it take if someone were to uncover a 4 billion-year-old fossil? What would we be looking for to say, okay, this is definitely early evidence of life?
So I think there's been a lot of arguments about fossils that are 3.5 to 4 billion years old. And really, it comes down to the question that Erlen was asking earlier. How do we tell if this is a physical abiotic process, or how can we tell that this is actually a true bona fide biotic process? And really, in order to do that, we need to better understand some of the constraints today about how do some of these physical processes occur and how they might mimic biological processes. And I think really the jury's still out. We really don't know enough about the physical world and how these abiotic processes could occur. So that's a huge source of the contention in that field as well.
Before we wrap up, I want to invite Judge David Tatel to come give some closing remarks. Judge Tatel and his wife and educator, Dr. Edie Tatel, have been instrumental advisors to the Carnegie Origins Project and to the Science and Society grant from the Carnegie Corporation, which made this evening possible.
So I am not a scientist, but I am, I think, a good listener. And I have 2 takeaways I'd like to leave you with. The first takeaway is that even though we invited all of you here tonight to learn about, quote, how and when did life originate on Earth, you may have noticed that that question was not answered tonight. In fact, our brilliant panel could not even agree on the definition of life. And this is my second takeaway. What you did see tonight, and this is what Carnegie Science and Society is all about. You saw an extraordinary example of the scientific process at work. That is, you saw how gifted scientists think about one of the most fundamental questions facing humanity. What are the origins of life? Thank you so much for joining us.